I'm a dermatologist at Mayo Clinic, and I've been working there for more than 15 years now. I trained at Mayo Clinic for dermatology. I was back after my postdoc in Rochester and thought, oh my God, what am I going to do now? And I thought, let's take a look at melanoma, because what we do in the clinical lab for melanoma looked pretty old school. You know, we're just basically measuring tumor invasion depth with like a ruler, you know. And I'm like, I just did quantitative proteomics. Maybe one could do something here, you know, to kind of refine prognostication. And that was one project. Also did other more basic projects, but this one project with melanoma kind of took off because it was perfectly suited for Mayo Clinic. Because at Mayo Clinic we are very well organized with like big tissue archives that go back decades, and everything's perfectly annotated. So it was very easy for me to have access to all these tissues and patient samples. And I started to do this molecular work. And then I was able to bring it to a certain point. So we were measuring, we were modeling, coming up with risk stratification models using different techniques. In order to show that the model has merit, you need to validate it, right? So you need to get these external validation cohorts, maybe even do prospective validation, and that is just a big ask for like a guy in a lab with a little bit of funding, you know. Should say all the research was funded by Mayo Clinic. Mayo Clinic owns all of the technologies that we developed, and they did a really great job supporting me, but still you come to a point where you need sort of industry support. And then, yeah, we found each other, Skyline, DX, and Mayo. But still, you know, doing these sort of specialized assays, it takes a special type of a lab and special type of infrastructure. And yeah, so that's what we kind of built with Skyline too, right? A lab that is able to sort of run these methods reproducibly and high quality. So how does Merlin improve our ability to treat patients? Before we had these types of molecular tests, all we had available to make predictions, diagnose things, are these stained tissue sections. So you get a recut from a paraffin block or wax block, you stain it with some dye, and then you look at that section under the microscope. And then you get sort of a set of data from that. People have researched that for a long time. You can do measurements like tumor invasion depth. You can look at tumor ulceration under the microscope. You can look at, you know, the number of cells that are actually dividing and those types of things. But then again, at a certain point, you run out of things to look at and to prognosticate and to use for prognostication and prediction. We know that there's sort of like a layer of information that underlies these phenomena like mitotic rate and tumor invasion depth. So there's a whole lot of sort of molecular change going on in the melanoma cells that you can't see just by staining a tissue section. You essentially take the tissue, and instead of staining the tissue with a dye, you extract the RNA, which is sort of like a message for the cell that tells the cell to make this particular protein, maybe an integrin, maybe something else, and then you can quantify that message and kind of deduce how much of this very important cancer-related protein is actually made in this particular melanoma. So it's a new type of an information that you can't see on the regular tissue section, and you can use it to then see, does it correlate with any important outcomes. So what do we worry about when we diagnose a melanoma? We always worry about the melanoma cells spreading to the lymph nodes, spreading to the lungs, spreading to the brain. So then the question is, if I know this information, can I better predict whether this melanoma is likely to do that? And that's a very important question because our subsequent therapy depends so much on the risk assessment. Yeah, there's a whole lot of, an entire layer of information that is sort of like hidden in the tissue that you cannot just see with simple slides in a microscope. You need to do molecular analysis. And that's what we did with Merlin. We screened, all the molecular markers that we could screen. We started with 16,000 plus genes that we screened. And then we found the most informative genes that would help us risk stratify, predict adverse outcomes for patients. And that's how the Merlin assay was created. And so when we developed Merlin, for example, we made sure that we used all the statistical tools that are available to make sure we don't overfit, that we don't model a noise, and that our model therefore has a high likelihood of generalizing to other populations, other cohorts. There's other tests in the market that have not considered that. They're huge mushroom models developed on tiny cohorts. And the likelihood that there's a lot of noise in that model is high. And in a way you can see it because these types of models tend to be very unstable and generate different performances on different cohorts. And that's exactly what we see, for example, with these other models. They validate very poorly. And in contrast, the model we built, I think it's just objective information. I think you can see it validates very consistently. So I think we've done this very, very rigorous statistical approach. We started that already at Mayo. We've continued that with Skyline. And as a result, we have a model that is robust and generalizes very well. Yeah, so how did we get to 8 genes and 2 clinical path features? Just exactly by what I explained, we used statistical methods to really extract the most informative genes, the genes that have the highest predictive information, and then matched that to the size of our development cohort, and came up with these 8 predictive genes and 2 clinical path predictors. And also for the standard of care clinical path predictors, we studied them extensively, and we found again that just by adding breast thickness and patient age, that this already gave you most of the performance of the clinical path predictors. So adding on additional clinical path predictors just gave you this sort of incremental, very small additional performance that then would have maybe introduced more noise than signal. And so we just basically ended up with these 2 clinical path predictors and married that to the 8 genes that were the most informative genes. And then we ended up with the model. So one algorithm versus many algorithms, or several. You can try to integrate the pathology information into risk stratifying strategy upfront, like we did. Early integrations, you have one algorithm that contains both the gene expression profile information and the clinical path predictors. You can also do it in a different way, like late integration. So you first, make your gene expression profile, and then you get an output, and then you have a second model that then puts other things into the mix. You can do it like that too. Competitor did it that way. The problem is a little bit like what I described. How do you make sure that your gene expression profile is not redundant with information that you already have in the clinic? Because we have extremely strongly predict, like tumor invasion depth, for example, is a very strong predictor of outcome. And so how do you make sure that you're not just rediscovering breast cell thickness with your gene expression profile, and if you then integrate things late, there's no additional sort of performance that comes from your gene expression profile over what you already have with breast cell thickness and these other variables. And so, we tried different things, but we found that this early integration worked best.